Now we get to start on how neurons work and send their signals. So the electrically excitable cells will communicate through what are called action potentials, or sometimes graded potentials. The action potentials are what we know as nerve impulses. So action potentials communicate through short and long distances, whereas graded potentials are only through short distances. Okay? So the production of one of these two potentials depends on the existence of a resting membrane potential. So in other words, the potential energy or movement the membrane of the neuron has available, and the existence of specific ion channels to change that membrane potential. Okay, so graded potentials are going to be short distances. Action potentials are longer. So here are some definitions, if you will. Resting membrane potential. So the potential of a resting cell. So nothing is happening in that neuron. Graded potential, a stimulus that will cause temporary and localized change in that resting potential. Action potential, stimulus that will cause an electrical impulse produced by some graded potentials that add up and create enough change for an action potential. So we will propagate along the surface of an axon to the synapse. The synaptic cleft activity. So neurotransmitters will be released from one neuron to the postsynaptic cell. And information processing, so response, so integrating that stimulus, response of the postsynaptic cell. We have a few channels that we'll see. So leakage channels. These are channels that stay open and allow potassium and sodium to move in and out of the cell. Ligand-gated channels. So we have to have a ligand or something that binds to the receptor to open that channel. Mechanically gated. We respond to a mechanical vibration or some sort of pressure to open the channel. Voltage gated, so a direct change in the membrane potential will open these channels. Here's another table for explanation. Okay, arresting membrane potential, so we are not conducting any kind of signal through that neuron, but we have a more positive outside and a more negative inside. So you can think of the membrane kind of like a battery. We only get that energy from the battery when we create a connection or we make a change to the battery. Okay, so to keep things positive outside, negative inside, we have the unequal distribution of ions through that plasma membrane. Okay, so more sodium on the outside, more potassium on the inside. Then we have most anions are not going to be able to leave the cell, so more negativity is inside the cell. And then we have sodium-potassium pumps that keep things equal because of those leakage channels. So here you can see more positive on the outside, more negative on the inside. So a number to make note of is negative 70 millivolts. That will be the resting membrane potential. Okay, so this just shows how there are going to be leakage channels. Potassium will move out of the cell, sodium will move in the cell, but these pumps, the sodium-potassium pumps, will pump sodium out of the cell and bring potassium back into the cell, creating that unequal distribution. As you can see, there are proteins that are negative This is what is causing the more negative charge on the inside of the cell. So graded potential, how we're going to change that membrane will be small deviations in the resting membrane potential. So a graded potential, that negative 70 millivolts, we can have some sort of stimulus and it changes it, depolarizes just a little to maybe negative 70, just enough to say something happened. So when this occurs, we have mechanically gated and ligand gated channels opening. You can see here we have calcium causing an opening of a channel. We have ligands attaching to a channel. We have other ligand gated channels. This will cause ions to move into the cell, but not quite enough to change the membrane to create an action potential. Okay, so the graded potential, we have different strengths that can possibly happen. As you can see here, we can add graded potentials together to try and really change that membrane. Okay, so what's happening when we are changing that membrane? The plasma membrane offers some resistance to ion movement. Again, unequal distribution, sodium chloride outside, potassium and negative proteins on the inside. Linkage channels will allow sodium, potassium to move in and out. Results, we need to create a resting potential, so those sodium-potassium pumps help maintain that unequal distribution. Okay, so an action potential, we're going to see a sequence take place where we depolarize that membrane. So we're going from negative 70 millivolts closer to 0. We actually move more positive. Then eventually we will restore that resting state, which is called repolarization. And action potentials are true nerve impulses. This will only occur if the stimulus creates graded potentials that reach the initial segment, or the axon helix, with enough energy to depolarize to threshold. So the big key word here is threshold, which is going to be negative 55 millivolts. So we saw how a graded potential can change the membrane a little bit, but if we actually add enough of those graded potentials to create a negative 55 millivolt membrane potential or state, this is our threshold to create an action potential. So this is what we have to reach to create an action potential. So that is seen here. So resting, negative 70. We have a stimulus. It is enough to reach negative 55. We cause depolarization, about plus 30. And then we start to repolarize. We actually dip down a little bit, hyperpolarizing, and then we level back out to resting state. The reason why we hyperpolarize is because we accidentally move too much potassium into the cell, and then we have to pump those back out. Okay, so this is all laid out. Depolarization to threshold. Activation of sodium channels. So sodium moves into the cell, depolarizing the neuron. Then sodium channels shut down and potassium channels open. This will start the repolarizing phase of the neuron. Then when things try to turn back to normal, we have a brief hyperpolarization because of the potassium channels not closing quite quick enough. And this will allow leakage channels to take over and the sodium-potassium pump to return the cell to resting membrane. Okay, these action potentials, the stimulus strength is not always going to make a difference. What I mean by that is to reach threshold, to create an action potential, we just have to reach negative 55. So if a stimulus is, let's say, just strong enough, so we just make negative 55, we still have an action potential. If our stimulus is strong enough to reach negative 30, we still have the same action potential. Now, if that stimulus holds on for a while, we can create multiple action potentials, but the mere strength of the action potential will not cause, the mere strength of the stimulus will not cause a change in the type of action potential. This is our all or none principle. So we either reach threshold and create an action potential, or we do not, and there is no action potential. Okay? So non-decremental means action potentials do not weaken as they travel down the axon. So once we start an action potential, it is the same along the entire axon. Okay, so this is just showing in pictures what is happening to change that membrane. Right. Propagation of those action potentials, we've got two types. We've got continuous and saltatory. So continuous is unmyelinated axons. So every region along that axon must depolarize. This creates slow speeds. Saltatory, we have myelinated axons. So this is allowing us to jump from node to node, or the space between the Schwann cells and myelination. This creates faster speeds. So continuous, we see each area of that axon must open up its channels to allow that action potential to travel. In saltatory, we jump from myelinated area to myelinated area. So this is going to allow us to skip sections of the axon, allowing for a faster propagation. Also making a difference would be an axon diameter. So the larger the diameter, the faster the speeds. We have less resistance. Again, amount of myelination. So the more myelination, the faster we are. Temperature. So the warmer we are, the faster the propagation. You can think of this, if you're cold, you can't really move as fast. You feel really stiff. So think of the same thing happening in your neurons. Signal transmission. So we have electrical synapses and chemical synapses. Electrical, we are transferring information. Think of smooth and cardiac muscles. Chemical synapses, we have a one-way transfer. So skeletal muscle, we are releasing a neurotransmitter from one cell to the next. These are different ways we can attach two axons. This is the synaptic cleft, so the junction between one cell and the next. Those neurotransmitters enter the synaptic cleft, attach to these channels to open them up, allowing for that action potential to move through.